Good morning, everybody. Lovely to see you all. We're just going to do a very little warm welcome, and Nick will obviously do the royal welcome afterwards. So we're just here just to do a few basic housekeepings for you. So there's lots of the RDS and the Dementia Research Centre team here today, and Helen's going to go through a slide shortly with everybody on. It's quite a gathering today, but we're all here to help and to support you, so please, at the lunch break and at the breaks, if you need someone to talk to, if you need any advice, any support information, please do speak to us. The toilets, if you haven't found them, are at the back of the room here. Ladies and gentlemen, if anybody needs any help with the accessible toilets, they're upstairs and there is a lift to go upstairs, so please ask one of the team. The emergency exits are through where you came in, and we'll go out to the front of the building. We're not expecting a fire alarm today, so if one goes off, please follow us quickly. We will be filming today. We've got the camera here. The filming is just at the front here, so nobody from behind will be filmed. So it'll just be the talks that will be filmed here. But just to let you know, just be mindful when you are asking questions, not to put any sensitive or personal information in. After the meeting today, we will be sending out a survey for some feedback. So please be as brutally honest as you can. Tell us what was good about it, what you didn't like, what you'd like for next time, anything which we could improve it. While we're speaking today, I've got a really loud voice, so I don't really need a microphone. But sometimes it can be difficult to hear. So if any of the speakers are a bit quiet today, please let us know straight away, because quite often this is the bit that comes through in the feedback was I couldn't hear properly. So please tell us straight away, and we can use some extra mics for that.
Okay, so as Nikki mentioned, there's just a couple of people here from the staff at the DRC and elsewhere. If I might ask anyone that's joined from the staff just to do a little stand up and do a little wave, if that would be okay when I get to you. So of course, you'll know most of these people. There's one there, that Nick, someone or other, who's helping out. So we have Dr. Natalie Ryan. We have Emily Goddard, who's done an amazing job doing lots of the organising today, so thank you. We have me, one of the nursing team. We'll be joined later, if not already, by Dr. Chris Hardy. We have Dr. Phil Weston here, Dr. Maniv Nazir, who's the clinical fellow in the local FAD team, who many of you will get to see if you've not already. We're joined by Minnie Beaumont, Dr. Xin Zhang. Gosh, there's so many people, I'm running out of breath as I go through. Sorry. Then we have Dr. Nadia Magdalinou, who will be talking to us about trials a little later. Emma from Rare Dementias Support, Emma Jones, who's done fantastic things with organising. Thank you to Emma. Nikki Zimmermann, who I think you might know. Ollie Hayes. Ollie, you here?
He's letting people in.
Ollie, who you might have met at the gate earlier. Then we have Professor Seb Crutch. Wave from Seb there, thank you. And then Tait, who's representing the National Brain Appeal. And then we'll be joined perhaps a bit later by Briony Campbell, our
artist.
I was going to say like resident artist almost. I'm not sure if that's the right term. Okay, a little recap on the coloured dot rainbow situation, as many of you will have kindly partaken in previous years. There will be an exam at the end to see if you can remember what each of them mean, so do take note. Of course, this is an opt-in system. If you've not got a dot already, there's a table just to the other side of this wall with them on if you'd like to wear one. The general idea is that it will provide a clue for you to connect with other people in the attendees that might be in a similar situation as you, to help with that connection element. So generally, yeah, go for someone who's got the same colour or colours would be my advice, rather than trying to remember all of the dots. This is a little recap on our agenda for today. You'll also have copies of the agenda on the chairs there. Of course, plenty of time for question and answers, refreshments breaks, and most importantly, a reservation at the pub after we finish. little slide here on our research table, which featured last year. If you would like to get involved and you're not already, or would like to do more in terms of research, then we have, or will have, our team just outside on the table the other side of this wall, talking about the different studies and providing information and how to sign up. That's the end of our slides.
And we're going to hand over now to Professor Nick Fox. Thank you.
Thank you, and welcome everyone.
Well, it's very lovely to see so many people here. Lots of people I've known. Some people I've known over a long time, and some people I've not met before. I look forward to meeting you very much, and I appreciate you've been amazing coming down in a very hot day. And I'm pleased that the air conditioning seems to be working, which is great. I'd like to start by acknowledging that for some people here, this will not be an easy thing to come along to, and it's... Do you need me to use the mic? Does anybody... Or is that because you're recording it, or does anybody have any trouble hearing me? No? Okay. I'm happy to use a mic. Can you hear me at the back? Fine. I usually can be heard. I've got one of those voices, Helen. What I was saying is that I do recognise that many people have come. Some people find it hard, and I really appreciate the people who've particularly made an effort, not just because of the heat, but because these things can be emotional and stir up things. But it's great that you're here. And the next thing I want to acknowledge is, while there are people with great expertise in psychology and in medicine here, you are the real experts. This is your meeting. You have that lived experience, which makes you the real experts. So that means that please do make this as interactive as possible. You will all have experiences and knowledge that some people may appreciate you sharing. I really value if you could feel completely comfortable asking questions, interrupt me, interrupt others, say that's rubbish, or whatever you need to do. But also, this is a special request, which is speak to somebody you've not met before in the coffee or lunch breaks or something like that. And so try and make those connections. I've always felt that when they very first started these meetings, we would have research talks. And then I realised everybody was much more interested in the coffee breaks and talking to each other. And that's entirely right and appropriate. So I thought I would do a little bit of an update. I will stand up here. But please do say if you can't hear me. I'm going to do a little bit of an update, but in time-honoured FAD annual seminar tradition, I will do, because some people will know everything about familial Alzheimer's disease, but I thought I would start by just saying a little bit about what familial Alzheimer's disease is. And I'm sorry for those of you who it's all old hat, but some people may not know, and there's a lot of ignorance out there. So I'm going to talk a little bit about that. I'll take questions about anything, interrupt me as you like. I'm going to talk a little bit about some of the research updates, and I'm going to talk about recent regulatory approvals and not approvals and things like that. So first of all, what is familial Alzheimer's disease? So this has lots of different names, and you may hear it referred to as very different things. So it's also called genetic Alzheimer's disease, it's called inherited Alzheimer's disease, it's called, and this is the mouthful, autosomal dominant Alzheimer's disease, or ADAD. They all mean the same thing. What they mean is that there is a gene that is causing Alzheimer's disease. These are very rare. You're very special. Do you know you're very special? But you're very special. And it is pretty amazing. I always find it slightly emotional when you see that there are people in this room who represent a big chunk of the total familial Alzheimer's disease population in the UK, all come together. Probably just for the coffee, but still. So Autosomal dominant Alzheimer's disease, we'll call it familial Alzheimer's disease, but it's the same thing. Those people who are involved in DIAN, the dominantly inherited Alzheimer network, they refer to it as autosomal dominant Alzheimer's disease. I'll explain what those words mean. Autosomal dominant means that it's not sex-linked. It doesn't just run down families, the men in the family or the women in the family. It means that there's just as much chance of inheriting it if you're a woman or a man. And there are great myths within families. People often, when I see people as a group in clinic, they often will say, well, yeah, I know all about this. And then when you see them one-to-one, they say things like, in my family, it's just the women are, so I know I'm going to get it. And that's just wrong. What happens is, there is a mutation, that just means an error. It's like a spelling error in a very complicated recipe. Unfortunately, the recipe is for something very important, a protein that doesn't give you any problems to start with, but over decades, it means Alzheimer's disease happens earlier. Instead of happening in your 80s or 90s, it might happen in your 30s, 40s, 50s or 60s, or whatever. So that's a spelling error, and that mutation, that's what it's called, that error in the gene, is passed down. And you inherit 2 copies of all these key genes, one from your mum, one from your dad. And they will have inherited 2 copies, and so back forth as far as you can tell. Let's say your mum was affected. She will have had two copies of the gene, one which was completely normal and was doing its best, and the other one that has an error in it. If you inherit the one with the error, then you are likely to get it. And you're likely to get the same disease at a similar sort of age. We used to say it is something called penetrance. That's another word that gets people to understand. What it means is how likely, if you have the gene, for you to get symptoms in your lifetime. And some genes, for other diseases, have very variable penetrance. So that somebody can have the gene and live to 100 and be fine, and a brother or sister also have the gene and be affected at 20 or 30. That's not the case with these diseases. And we used to say it was 100%. That if you got the gene, you would become affected if you lived long enough. And I say used to, because there's been a few amazing discoveries recently which suggest that very occasionally, rarely, you can have a cocktail of other genes that you've inherited that somehow compensate. That's not to say that if you lived to 140, you wouldn't still get it, but it disrupts that if your mum got it at 60, you're going to get it at 60 link. And anybody who knows about their family will know that even though we say that this is inherited with a similar age at onset, similar age when you get symptoms, it's very variable within families. I mean, isn't it? I mean, people know that there's a difference. And that's somewhat being ignored, and there's work that Natalie's doing with DIAN, with a big consortium, to try and understand what these other genes are that mean that in one family somebody will become affected at 55, and a brother or sister might become affected at 65. Of course, it's very similar age, but those 5 or 10 years makes a big difference. And that brings me, I'll talk about what genes there are in a second, that brings me to why you all are so incredibly valuable. No comment on your personalities or anything like that. This is just that you collectively, globally, particularly in the UK, have given so much to research that has transformed our understanding. I mean, truly transformed. And I'll explain that in a moment. But this idea of understanding what genes might affect how soon the error in the gene will cause problems, whether it could be delayed, will be crucially important, because if you find something that from these other genes that are delaying things, then you can make in a laboratory whatever that gene is making, and then that becomes a treatment. And I think people who in these families, I know that, you know, delaying an onset by 5 or 10 years, well, that's not a cure. I think more people would really take that, wouldn't they? So what are the genes that can cause this? There are 3 main genes. One is called APP, one is called, which is amyloid precursor protein, and the other is presenilin-1 and presenilin-2. And presenilin-2 is not that common in the UK. Presenilin-1 is the most common, and amyloid precursor protein, or APP, is the second most common. There are some very rare other things as well, but those are the 3 main genes, where a single error in that gene, any one of those genes, will cause Alzheimer's disease. Usually early onset, but not always. So amyloid precursor protein was the first gene, and it's all in the name. It does what it says on the tin. Amyloid precursor, well, the beginnings of protein. So that gene codes for a protein. It's a huge protein. And a little tiny error, one error in the building block of that protein, makes amyloid, which is its normal production, the little protein, the little peptide, just a little bit longer. So normally we make this little protein, which has got 39 or 40 building blocks. And this error means that you get 42 sometimes, rather than 40. Not all the time, but when the body is chopping up the amyloid precursor protein, sometimes it's slightly longer. And you thought, well, how can that make any difference? And what it does is it makes that amyloid protein stickier. It sticks to itself. And actually, when we developed blood tests, one of the people had great trouble with blood tests and with the cerebral spinal fluid tests, because the protein is so sticky, it stuck. People didn't realize things weren't working because the protein was sticking to the walls of the test tube. And so they thought there wasn't any protein in there, but it was stuck to the walls of the test tube. You used the wrong test tube, and it looked like there was no amyloid. But as a result of it being stickier, it sticks to itself. You've got one little protein swimming around outside your brain cells, it gets cleared. If they start sticking to each other, they very quickly go from one stuck to another, so 2 to 4 to 6 to 12, and then the brain can't clear them. And that process takes years, and the clearance basically is overwhelmed by the fact that this is more sticky. The other gene, presenilin-1, which just means early onset, pre-senile, is the key, codes for the key enzyme that chops up APP. So it all came together in a story that's understandable. That amyloid is key to causing this illness. And that for decades was debated. And families like yours gave the huge understanding. And when everybody debated experiments on mice and things like that, they said, well, look, you cannot get away from families are living evidence that this is important. And that led to us understanding that there was a pre-symptomatic phase of the disease, actually 20 years of amyloid gradually building up. And it led most remarkably, and I think people in this room or their families and families like yourselves around the world should take credit for it, led to the idea that if we could remove amyloid, we could help the brain remove the amyloid, maybe that would help people. And maybe that would help not just people with rare mutations. You're far less than 1% of all of Alzheimer's disease, but obviously you're more important. Maybe it could help the other 99%, and that's exactly what was found. And we did here the very first vaccination study with one of the sites, where we vaccinated people. Somebody had this malady, you vaccinate people against amyloid, maybe that would help the brain in clearing proteins, clearing the amyloid. Recognize it as foreign, just like you get vaccinated against measles or COVID, and you remove it. And that caused dreadful side effects, inflammation in the brain, because as part of removing it, it caused inflammation. The inflammation was part of the extracting it out of the brain. But now, after 25 years, we now have 2 drugs that clearly remove amyloid. Amyloid that's built up over 20 years gets taken out of brains of people in your families or without a family history in about 18 months. So that the majority of people in those trials had so little amyloid in their brain that they wouldn't have been eligible to go into the trial in the first place. Which is remarkable. They wouldn't have met criteria for Alzheimer's disease trials. And everybody said, oh well, we've taken amyloid out before and people haven't got any better. Who cares if your amyloid's got out your brain but you're not slowing the disease? And the answer is, you had to really, really remove it. Removing it a bit, which is what the early treatments didn't cut it. And it had to be reduced hugely. And now the first drugs. And you will see all sorts of things in the press. And there are people, at times I think that people don't want treatments. I just find it hard to understand. These work. They are not wonderful. They're not cures. They have side effects, but they work. They slow progression, one of them by about 27%, a quarter, and the other by almost 35%, a third. And it's clear that the earlier people get treated, the milder they are in terms of how long the amyloid's been in their brain and whether they have symptoms, the better they do. And that is a remarkable breakthrough. And like the first treatments for infections, the first antibiotics, being told by medical historians that they were absolutely rubbish. The antibiotics made people feel really ill. They did things like, I think they re-purified antibiotics from people's urine because, if you had the antibiotics that were so valuable, you had lots of side effects. They've got better and better. We just expect that we can pop a pill and it will cure our sore throat with antibiotics. And the same is true for cancer. The same is true for HIV. They weren't very good to start with, but they rapidly got better. And what the big delay is the first treatment. That is opening the door. That is when things accelerate. And partly they accelerate because companies, which is where the money is and where the effort can be put into, believe it's worthwhile because they believe we can do this. And there are a whole range of treatments coming through now where what's clear is that these treatments need an intravenous infusion, so a needle in your vein, takes half an hour to an hour. Apologies, Nadia, if you were planning to say some of this, but maybe you'll be talking about other things.
Yeah.
Repetition's good. But so that's not straightforward, particularly if you live a long way from a centre like ours. You might have a lot of travelling. But also, between about 12 and 24% people get a side effect, which means they have to stop taking the treatment or pause, or they can become unwell. Most of the time people are not. 80% of people who have this side effect don't have any symptoms. And it is again inflammation in the brain as your antibodies, which is what the body makes naturally in response to an infection, say, that you give them the antibodies in these two drugs are antibodies that attack amyloid. They're anti-amyloid antibodies. And they help the brain get rid of it. Bits of inflammatory cells gobble up the amyloid and they get cleared. And there are pictures of these cells with amyloid inside them, consuming them. It's sort of kind of wonderful. It's like sort of see, the fight back. So the next treatments that are more hopeful in terms of being less burdensome, there are really promising treatments which are just subcutaneous, under the skin, like people give for insulin in diabetes. And then a remarkable treatment which only just was presented back in March, which is one of the reasons why you get these side effects, get the inflammation, is because the brain has a barrier around it to protect it from infections and other things. And so it's very hard for the antibodies, the drugs, to get in. So you need quite big doses, because only less than 1% of the drug that's coursing through your veins gets into the brain and it can attack the amyloid. and then really clever, what's called a shuttle technology, where something the brain normally takes in and out has been hijacked, and you put the drug onto it, give it with this shuttle, and it gets into the brain, amyloid is cleared even faster, and it doesn't give you the side effects. So those things are great hope. Okay, in my last minute, where are we with these two treatments? I feel they're a landmark, they're a great source of hope. They're not perfect, they're not a cure, but these antibodies will remove amyloid. There are trials which Nadia will tell you about, which are really exciting, but I'll tell you about the realpolitik of licensing. So the FDA, the Federal Drugs Administration, the Food and Drug Administration in the States, license these drugs. And then slowly, Japan, Korea, and then finally the European Medicines Agency approved it for Europe. Long processes. Are they safe enough? Do they give enough benefit? And I've been in a lot of the European Medicine Agency meetings, and honestly, it was a battle. And I don't really know why. I think people are frightened about new things. You hear side effects of bleeds in the brain. These are microscopic amounts of blood. They don't cause any harm. Inflammation is very frightening. Maybe there's also a worry about so many people, because Alzheimer's is so common, overwhelming health systems. I don't know. That's for the coffee break to talk about that. But finally they've been approved in the UK. Not for everybody, but for most people. There's a particular genetic variant that everybody has, that if you have that, you're more likely to get the side effect, and they're not eligible. And very sadly, a couple of weeks ago, NICE, which is the body that decides whether it's affordable by the NHS, turned it down on its third appeal. And again, I'm happy to talk about that, but in brief, I honestly think it wasn't malicious. I don't think it was, because people have said, well, these are expensive drugs. They cost £20,000 a year. It'll overwhelm the NHS. They can't afford it. There are hundreds of thousands of people with Alzheimer's disease. Well, the answer is they're only eligible at the very earliest stage. There wouldn't have been hundreds of thousands of people coming forward each year. There would be thousands, but not hundreds of thousands. That's to start with. So it wouldn't have overwhelmed the NHS. And they were quite good at weighing the cost. How many extra brain scans? What is the cost of an intravenous infusion? But what they were very poor at doing, because they've got great expertise in heart disease, in cancer, of saying this drug will save 2 years of life. Therefore, costs this much pounds, gives you 2 years, they have a formula. And it comes down to, I'm afraid, as simple as, is it more than £30,000 for every extra year of full quality life you get? Less than 20,000, you're almost certainly going to get approved. Up to 30,000 perhaps, beyond that, you won't. And that is to protect the NHS, and for good reason. But what they couldn't do, in my view, because they didn't have the instruments, they didn't have the experience, is weigh up the benefit to families, to wives, husbands, partners, children, as well as to the individual who has the problem. The idea is that in Alzheimer's disease, we're not just looking to give you an extra year of life, but it's to try and keep people when they are well. And that is a battle that we will continue to fight. But we need you as advocates. We need you to help with research. This would never have happened without you. It wouldn't have happened without finding the gene. And when I had my very first conversations 25 years ago with drug companies, and I said, look, the best people you should treat, the people with these families, they will give you a limb easily. They'll say, which one do you want to try and find a cure? They're well, they're young, they're strong, they haven't got lots of other illnesses. And they know what has happened in their families. They are the most motivated people you'll ever meet. And they said, there are not many of them. And maybe they're not typical like Alzheimer's. I said, they're not typical, but there are more than you think out there. I said, well, I don't think we could run trials. So they refused, and they even had age limits, so that people couldn't go into the standard trials. You had to be over 55 for the first trials, which just excluded the most motivated people you could ever imagine. And what they said is, show us that there are enough families out there that will come forward. There are chunks of the UK where there hasn't been a single family having a genetic test. Now, I don't believe it's the drinking water up there. It's just that doctors haven't done the tests. So you need to spread the word, because that is in your own self-interest. Of course, we're keen to be part of that, but you need to get other people, members of your family, to come forward, sign up for research. We won't take any limbs today, but we have a project, don't know. But joking apart, you are incredibly valuable, and you have contributed so much. And so the two things I want you to take away is that there really is hope. It won't be in time for everybody, but what you collectively have done have transformed Alzheimer's disease treatments. Thank you very much.
Thank you, Nick, that was fantastic. And I'm delighted to introduce Nadia, who's going to tell us all about trials.
Hello, thank you for inviting me to talk today. I'm one of the doctors working with cath memory in the trials unit at UCL and UCLH. Nick covered a lot of what I was but he essentially, I wanted to say how the whole community, I see people in clinic who are upset with NICE not approving lecanemab and donanemab, the two anti-amyloid treatments, and also the geopolitical situation is not ideal at the moment, but there is, I'm here to give...
Nadia's another person who has a great voice. Can you hear? I just realised that this is pointing away from you, Nadia.
Okay.
But
can you hear me?
Yeah. We only allow people to work. They have to check at the Dementia Research Centre. They have this voice projection test first. Otherwise.
And also, on a personal level, I'm a jaded Greek, so I'm usually not optimistic and not, glass, half full. But I am very optimistic about what's happening in the field, and I'm very excited to be working in it at this specific time. So what's next for the dominantly inherited Alzheimer's network trials? So first of all, this is a very exciting slide. That's only in 2025, and you can see that in the drug development pipeline, there are 182 trials and 138 novel drugs. That is amazing. Even a few years ago, that would be, fantasy. And that's really important because I'm sure you've seen this graph before in some form or another, because it shows people in the FAD group at different sort of stages, different cohorts. So initially you have primary prevention, and I'm going to talk about a really exciting study we'll do in that group later. And that's people who do not have any biochemical evidence of Alzheimer's disease and who do not have also any symptoms. And that's a group that hasn't been explored before. And in the middle, you see the secondary prevention group, and we have 2 trials in that group. And that's a group of patients with some biochemical evidence of Alzheimer's disease, most times without any symptoms, or with mild symptoms. And we have 2 trials in that group, giving medication to sort of slow down and sometimes prevent the onset of disease. And the last group is a tertiary prevention group, and that's people really we see in clinic who have sort of garden variety of Alzheimer's disease, sporadic Alzheimer's disease. And they are people who have both biochemical evidence of Alzheimer's disease and also have clinical symptoms. And that's the 2, so as Nick said, the lecanemab and donanemab, the 2 treatments, anti-amyloid treatments, they were for that group. So just apologies if you've seen this before, targets in Alzheimer's disease. So we talked about the accumulation of amyloid plaques, and these anti-amyloid monoclonal antibodies essentially are like hoover up amyloid that is already in the brain. Another significant target is hyperphosphorylated tau, and we know that correlates very well with disease progression. And there is another drug that I'm going to talk about, E2814, which is a monoclonal antibody for this particular protein. And it's a complex process. There is a complex interaction with inflammation and neurodegeneration. I'm not going to talk about other trials that we do targeting inflammation, but I'm putting it there that this is, it's not a straightforward. That's why we're not quite there yet, but we're getting there. So talking about, you've seen this slide if you came last year about the secondary prevention trial with GANT that has happened already. It started in 2012. It is really important because it was the first time dominantly in FAD an anti-amyloid treatment was used, and it showed that those two drugs are safe, which is great, but unfortunately didn't show any clinical benefit. So then researchers thought about it and said, what about if we increase the dose of anti-amyloid treatment in GANT and we give it for longer? And we got really exciting results, fresh off the press in April 2025, that showed for the first time that long-term and importantly full amyloid removal. So as Nick said before, it's not partial what we're doing before, potentially delayed symptom onset and dementia progression. So in the FAD group, in the FAD population, 22 out of 73 participants who began treatment before cognitive symptoms and received the drug for an average of 8 years, half the risk of developing dementia. And that is extraordinary. So the best results were in the group reaching near normal amyloid levels or who were treated many, many years before symptom onset, sorry. So then this is exciting, but where do we go from there? So we need further research to unpick what this really means. And that's why we now doing another trial called amyloid removal trial that has started recently. And this examines the effect of continued removal of amyloid on disease. And instead of giving GANT, we're giving the lecanemab, an infusion that is given every 2 weeks. Everyone who's participated in the open label extension is eligible. So we have mildly affected individuals and asymptomatic individuals. And they must be co-enrolled in the observation arm of the DIAN study, and Monique has been amazing helping with that. It's long duration, so we've learned from the past, so 7 years, and they can be home dosed, and we have experience with home dosing, I'll discuss later. This has been really tricky to set up, and we thought at some point that we wouldn't be getting it off the ground. But we have our art angels in Helen Rice and Jane Douglas, who wasn't able to join us today. They exceeded everyone's expectations by recruiting participants within 1 week. And it's a typo, or my computer couldn't believe it, that they actually managed to get people to go to the US for amyloid scans, and Spain, which is, I share an office with Jane, and I know how difficult it is. So we've consented to individuals already. Monique has done 2 LPs as part of the ops arm already, and we were going to dose the first patient next week, but this has been postponed because of scan issues. But we're getting there, and Helen and Jane have been absolutely amazing. So talking about another immunotherapy, but tau immunotherapy. This is very exciting because there is a collaboration between UCL and ESI and industry, and this is what happened? Yes, this is very valuable in terms of industry has the money, sort of academia has potential mechanisms, so it's good to work synergistically. And previous DIAN-TU antibodies went successful, but E2814 targets tau well, and it's safe. So this has been used as part of another prevention trial, DIAN-TU 002, and we have, I'm glad to see some of the participants here. Jane has also been amazing in coordinating that. So this is the first trial that we used lecanemab, the anti-amyloid treatment, and also E2814 or placebo. So we have 2 different cohorts. So asymptomatic participants get E2814 or placebo for 1 year, and then they get the anti-tau treatment, and then they get open label lecanemab, so they definitely get the anti-amyloid treatment afterwards. And for the symptomatic cohort is the other way around, so they get the open label anti-amyloid treatment for 6 months, and then E2814 or placebo afterwards. And these are a lovely research nurses supporting that, Anna and Alex, and that's at the infusion suite at Elwick, our experimental center. So this is really important because our primary aim is to understand the biological effect of tau treatment, and it's also sort of going back to the previous sort of cartoons I showed you, adapt to the changing clinical landscape. So we have more treatments. We know there are several mechanisms that could, are involved in Alzheimer's disease, and what is, our best chance of success. And as Nick said, also learning from different diseases, it's very rare that someone who has cancer gets one drug. It's a combination of drugs that are needed depending on what stage of cancer they have, what other comorbidities they have, so what side effect profile of medications they would be able to tolerate. Unfortunately, there's no free meal, so if you include 2 medications, the complexity of trials increases, and as is safety concern, how to disentangle what is causing what. The duration is longer, 4 to 7 years, and we've successfully home dosed, I think, 6 out of 10 participants in this trial. So there's another, so we talked about so far about antibody treatment. So this is another collaboration between academia and industry, led by Dr. Ross Patterson. And this is a gene silencing treatment. So this is a treatment that interferes with the tau gene, and then decreases tau levels that way. And at the same time, they are looking, and Aram has been fantastic at recruiting patients and completing the first part of the study. They're looking at a method called SILK, seeing real-time changes in tau production and clearance. What I haven't told you is that when we collect data on patients participating in clinical trials, we also increase the understanding of the disease processes, because we're collecting lots of different biological samples, and usually people also consent for future research. So that's invaluable information we get hand in hand with whether a drug is safe or effective. So Aram recruited 5 participants, 2 with mild to moderate FAD and 3 with sporadic AD, and they've completed the first part of the study, and he just, which was placebo control, and he just dosed the first open label participant, who will definitely get the active drug this week. The WashU will start screening anytime soon, and they're aiming also to include 5 participants, 3 with FAD and 2 with sporadic Alzheimer's disease. So that's the last medication I'm going to talk about. Nick talked about amyloid precursor protein, and this is another very exciting drug. It's a medication that again works with, it's a gene silencing treatment. It's slightly different mechanism to the previous one, but it essentially means that it turns off the tap. It turns off the tap of amyloid production, and as you see in the graph on the right-hand side, we can see in the cerebrospinal fluid of participants that APP levels decrease and amyloid levels also decrease. This is a safe drug. Sorry, my IT. This is a safe drug. We've recruited really well. We're only the second center in the world. Only Canada has recruited more than us. We have 8 participants, and we have one of those participants has confirmed FAD. And it's really important because we haven't seen any evidence of the areas of small bleeding or swelling in the participants, and that is excellent news. So as we learn more, we can sort of identify patients who would be at the beginning, who would be at higher risk of these bleeds, and we would give them this medication rather than other medications who would make the risk of hemorrhage and edema and swelling higher. So that is very important. And finally, talking about the prevention trial, so primary prevention. So this is again the first time that would be done. The goal is to prevent amyloid plaque pathology and disease onset, and it's an anti-amyloid medication that is given subcutaneously, which is also very important. So the estimated years, people are going to be included 11 to 25 years before symptom onset. And the enrollment is to start in September 2025. It has been tricky to, take it off the ground, but it's happening. So there are 2 parts. So the first one is placebo-controlled, with very little amyloid. I'm really sorry, I don't know what's happening there. At entry, there's a 50% chance of getting the medication or the dummy drug, placebo, and for a year's duration. And the second part is the open label extension, where everyone is going to be getting the active drug and another 4 years duration. So to conclude, there are multiple trials ongoing in FAD. There are, of course, challenges, so people need to know the genetic status to participate. There's frequency of dosing. People have been coming initially for the lecanemab infusions every 2 weeks, which is tricky, and obviously side effects we discussed. But there will be multiple opportunities to participate in trials, and new therapeutic opportunities come up all the time. So there's lots of exciting things happening. And that's my last slide, I promise, or my penultimate slide. I just, again, reiterating what Nick said earlier. So on the left-hand side, you'll see a trial assessing donanemab on the anti-amyloid treatments, and it's admittedly modest effect, tracking, cognitive improvement in the group getting donanemab versus the placebo, the dummy drug. But on the right-hand side, you can see AZD, which was the first antiviral medication to be approved in the late 80s, FDA approved for HIV/AIDS. And there is a similar effect. There was a lot of, in the medical community, people were not convinced that was the right decision, and there was a lot of controversy because of the modest effect, because it was given intravenously, because it was, it wasn't perfect. But that was the first step to HIV/AIDS becoming from a death sentence to a chronic condition, at least in the countries that can afford the medications. And also, it's no longer used because it was the first blunt instrument. I looked it up yesterday, and now it's given in an oral or syrup form, so it's no longer given intravenously. So we will get better, and this is an amazing. The 2 new anti-amyloid drugs are amazing first steps. So this is my last slide. So I wanted to say that. There is a saying that in order to bring up a child, you need a village. I think in order to bring up a clinical trial participant, you need a whole country. So we're very lucky to be part of an amazing team, and we're very grateful to our sponsors, but we wouldn't be able to do any of that without your participation, and you know, our participants are amazing, and the families supporting them are amazing. So thank you very much.
And just to say, we're going to have a final panel discussion at the end of the day, where anything that's come up during the day, there'll be more chances to ask questions about anything and discuss. And this does lead in really nicely to our next talk, which is from Briony Campbell. You were described as our resident artist earlier, which I think is a really, if that's okay, a really wonderful way of describing your involvement in our community. So a few years ago, there was a call sent out around UCL for people who might want to apply for projects funded by UCL Public Art for what are called knowledge exchange projects for scientists and clinicians to partner with artists and partner with the people from the communities they work with to develop creative projects with the ultimate aim of creating artworks to go in the new Translational Neuroscience Centre that's been constructed for UCL. So obviously I thought I have to apply for this. I remember Nick saying, be careful, these things can be a bit of a distraction, but actually it has been the most amazing distraction, because this project has taken us down so many new routes that I couldn't have possibly imagined it would have, in terms of raising awareness of familial Alzheimer's disease, because of the amazing people who took part. And we wanted our artworks to be very much a part of the fabric of the new centre, and for your stories to be the things that people came to that centre would be connected with. And so I was lucky to meet Briony, and we were lucky we put out an open call for anyone who wanted to get involved to join our team. And you're going to tell us a little bit about what we created.
Yeah, thank you. I was told I should stand on that side to have a better chance of the screen working well. Hi everybody. So I'm really pleased to have a slot here today, because I know you've got huge amounts of science and important things to get through, but hopefully this will be a little...
Just with the microphone.
Can people hear?
Oh, sorry. Should I stay on the microphone?
You can take another mic.
What should we prioritise? Oh, okay, a roving mic. Okay. I can stand on the stage.
Oh, it's fine.
Okay. Okay, thank you. Is that better? Okay, so Natalie gave you a little intro to the beginning of the project. We were invited to a matchmaking session where lots of artists came and lots of scientists, and specifically neuroscientists in this case, wasn't it? And then you had to see if you met somebody who you liked the look of, and I liked the look of Natalie. So we bonded over. Sorry, the slide's not quite right. Okay. This is the title of our project, Walking in Your Footsteps. And we bonded over the fact that Natalie brought along the letter is this something that everybody would know about? Yeah. The very first letter from the very first family who suspected that there was an inherited gene in their family, and sought advice, which led to recognising the gene ultimately. And the fact that Natalie had brought that along, we were all asked to bring something that would represent our practice, a little anything. And the fact that she brought this along really showed me how much she valued all the families like yourselves, and I really loved that about her. She didn't I'm very unscientific myself, and she didn't kind of boggle my brain with complicated, impressive science. She just went straight to talking about the families. So that was the beginning. So we began to build a proposal for a project that we might do, and these were our goals. To learn more about the lived experience of families affected by FAD and the stages they go through. To offer participants an enriching experience and connections with others from families affected by FAD. To improve awareness of and support for families through sharing the new perspectives we co-develop. And finally, which is yet to happen but will happen, install artworks in the new research institute, which would provide an enduring reminder to the scientists who work there of the immense contribution that families affected by FAD continue to make to our understanding of Alzheimer's disease. And those artworks will also be seen by all of you and anyone who attends the clinics. So with that mission in mind, we had a call out to your group, and I've got some faces in here who are participants who joined in. There's a few of them, and we began with a collage workshop. And the idea of the collage workshop was really to generate conversations. It wasn't so much about making a final outcome, but the process through which we did it was sort of through prompts where we would say, okay, take something, we had a pile of stuff on the table, take something that could represent how you felt when you first heard this gene was in your family. And we'd put something on the collage, and then go through more questions that would help people to either talk about it or just think about it. So there was sort of no pressure to talk and not much pressure on the art, but whichever suited you better to kind of start expressing and thinking was the way that you would go, and it very quickly generated conversations and bonds. And I don't need to speak for the participants because I've got two lovely responses there. I'll just let you read. Or should I read them? Yeah, I think one of the biggest effects of the whole project throughout was this idea that the group got to spend that time together, where usually, like, all of you might meet at this event and there's a lot going on and small chances to chat, so to have that chance to actually be together and really get to know each other, I think was hugely valuable. So recognising that was a key, we went further and took everyone to Margate for a weekend. Oh well, first I will show you this image, who came from Tamara, who some of you might know. She brought along to the first workshop. We invited people to bring their own family photos. She brought this picture, which is of her dad, and he had a tradition of drawing beach mazes for them. And this really struck us instantly as a perfect analogy for what you might go through living with FAD and the constant looking for routes through, looking for solutions, hitting dead ends, trying to find the joy in the struggle as you do in a maze, but inevitably the tide is washing this maze away. So we thought, could we recreate that? It would be a perfect piece. And at first it was a completely over-ambitious idea to go to the beach and do that, but basically the participants all said to me, you've got to go and persuade the commissioners, you've got to find the money because we want to do it. So against the odds, we did, and we took everyone. This is the final film that is Tamara recreating a maze. And I'm going to play that. If anyone would like to see it in full, I'm going to play it at lunchtime, just on a laptop outside. And this is our trip to Margate. And we had sea swimming, and we wrote words in the sand, so people wrote words that were significant to them, perhaps from their loved one. That one says, today I start to fade away. Today is the day I start to fade away. And that one says, sorry. So these are different people's words. And we had a bonfire at the end of the night, which was amazing. Yeah, and so when everybody came, we had a sound artist come with us who drew this beautiful artwork. So we were also working with the theme of fingerprints for obvious reasons. Fingerprints represent DNA. They also represent touch and love and connection. So working through these ideas, so we turned a fingerprint into a maze, and then people helped her construct it, and walked through it, and that was a very sort of ritualistic encounter. And then this is, some of you might know that we recently had an exhibition, hopefully some of you came, of another outcome for the project, which was this series, which is a series of small images in Petri dishes. These are glass Petri dishes, and the idea to put them in Petri dishes came from the group saying that they really wanted the scientists to understand that the cells that they study are real people's lives, and there are real stories behind that, because some of the lab scientists don't get a chance to meet with families and really know about their experiences. So again, a very direct way to bring the two together. The images, most of the images start with people's family photos, and then I sort of handwork them to add ideas and themes, and this idea of lines of connections came up a lot. They could be bloodlines, they could be red tape, which is a huge element of all of your lives, I'm sure, navigating finding care. And then also we noticed that this idea of red tape, which is what you have to deal with, well, the motif of red, Natalie said that she felt that you all should be having a red carpet rather than red tape because you're so important in research. So we thought these red lines could be the positive or the negative side. So they came up a lot. And yeah, I should probably wind up, shouldn't I? I brought a few little pieces to show you out in the hall. And there's just one last thing I wanted to say. If there's one more piece of artwork we're going to make, which involves fingerprints from the group. It's going to be a big screen print, and if anyone else would like to contribute a fingerprint, I've got some ink, and at lunchtime all you have to do is come and push a fingerprint, and then your fingerprint will be part of the next artwork. That's it.
Proactive member of our group. Our May's workshop was featured on BBC South East News, so we really did reach a very big audience in terms of what we were doing, and actually the artwork that you've created I now use in all of my talks. So when I gave a talk last week to train junior doctors in neurology, and so I think it's just really just helps them to see what the experiences are when you have familial AD in your family, and I think it hits a level of resonance that maybe some of the scientific slides don't. You see people nodding off in the back of the room, but they were all extremely engaged in this talk, and I've given that talk in Turkey to the Alzheimer's community there. So it's going all over the world, and actually the DIAN team, I sent a little film that we made of our footage to them, and they're showing that in their waiting room, and it's on their website as well. So we are getting our messages out there. And really that leads on to awareness raising and fundraising. I'm so glad that Eva could join us for the National Brain Appeal. Oh, yes, so. The next one.
That's it, yeah. Thank you. Hi, I should say that I have seen Briony's piece, and it's really wonderful. So if you do get a chance to see that when the new Home for Neuroscience finally opens, then I hope that everybody will get a chance to have a look at that. It's great. My name's Eva Tait, and I'm the Major Appeals Manager for... quite sure what's going on. I'm not Emily. For the National Brain Appeal, and we're the charity that raised funds for the National Hospital for Neurology and Neurosurgery and the Queen Square Institute of Neurology as well. We've been supporting rare dementia support since the early 90s, and actually even before that as well. We've been raising funds through a variety of different ways each year, and we've also led on a fundraising campaign which raised nearly £10 million for the Rare Dementia Support Centre. So hopefully this meeting next year will take place in the Rare Dementia Support Centre, and also to pick up on something that Briony said, that will definitely be a place which will provide space for everybody to meet a lot more often, get to know each other a lot more often in a much more informal, comfortable, non-clinical setting. And having been in there last week, work is very definitely moving ahead on that. All the spaces are opening up, and it really looks to be the most fantastic building. So we're all delighted that we've managed to achieve our fundraising campaign for that, and that is in no small thanks to the RDS community, a lot of whom have done a lot of fundraising. I can see somebody over there who ran the marathon for us, Phil. So I'd just like to say thank you to anybody who has been involved in fundraising in whatever way you have. There's lots of different ways that you can get involved. You can just sign up to our RDS e-letter, which is great because it helps us to spread the word about awareness raising as well as fundraising. You can take part in events and challenges, all the kind of physical challenges. We had somebody incredible, age 74, do a wing walk. So if you follow us on social media, you'll be able to see a little film of a lady strapped to a plane. I don't suggest everybody does that, but there are many different ways you can get involved, and I would encourage you to come and talk to myself or members of the National Brain Appeal about any ways that you have, that you're interested, or your extended family and friends are interested in fundraising. We also do, we raise funds as well and take donations in memory as well. So there's other ways that you can become involved there. So please do come along and have a chat with me. I'm here at lunchtime. I've got lots of materials and I can talk to you about anything you'd like to talk about to do with fundraising there. One person in particular I'd really like to highlight is somebody called Bernard Bunting, who I believe has just passed through Edinburgh. He's on a very fast route south at the moment. Oh, we've lost him. He'll be back. So Bernard was diagnosed with early onset Alzheimer's, and he is taking part, or has been taking part in a clinical trial.
Still is.
He still is, yeah. But yeah, he's an incredible human being. He is cycling well over 4,000 miles around the coast of Britain he'd like to raise £1 million for the Rare Dementia Support Centre. I believe he's about £25,000 in, which is incredible. I think all sorts of people have donated to him along the way. I would love it if people could share his story, because that's really his original idea, was that if everyone shares it and everyone just gives £10, that would get him to his £1 million. So he's getting there. He's got some way to go, but he's getting there. So please do, if you follow us on social media or follow RDS on social media, you'll see information and posts about him. He is due to come back into Queen's Square, we hope, sometime towards the end of this week, and possibly Professor Fox and Professor Crutch might accompany him, depending on which day he makes it back in. So I believe he's going quite quickly, so it might be sooner than we think, knowing him. So, we have incredible members of RDS who help us raise funds, and he is a prime example of that extraordinary support. So thank you to everybody who's done it, thank you to everybody who'd like to, and even if you can just share our message and share that the National Briony Campbell support Rare Dementia support, that's really, really helpful to us as well. So do come and have a chat to me at lunchtime. Thank you.
I'm going to share some studies that we've literally just found out in really the last few weeks and months that have been funded. They're very early stage, but I think actually it's quite nice to know what's a bit further ahead. And these are things that absolutely build on everything that people in this room and their relatives have contributed over the decades that have preceded and led up to today. Today is our sort of 15th anniversary. We set up the support group back in 2010, and Seb, Nick and I, we were a very small little group then. It was before Rare Dementia Support as an umbrella organisation existed, and people in this room really helped us get off in those early days, making our newsletters, making our website when it was in its early phases, and we wouldn't be here today without all that. And that's sort of why we have this image of the dragonfly. So, and you've heard a bit about the Walking in Your Footsteps project, this really lovely art collaboration that we've been involved in. And through all the conversations that happened, and in that the dragonfly became important as a symbol really of the people that very much love parts of people from our community who are no longer with us, but have created what we have today, and who are still working really hard in their contributions to research through the donations they've made. So the blood samples, the MRI scans, the skin biopsies you heard about, and in some cases through brain donation. And the dragonfly has been a really important symbol through lots of cultures and through history of regeneration, of wisdom, of hope. And those who were here last year will remember that Sarah shared this story, the dragonfly story, at the end of our meeting as a story that she'd found really helpful for talking to her own children after her dad had died from familial AD. So this is here if anyone wants to have a look at it. And through our conversations with the sand artist who worked with us, we learned that the dragonfly was an important symbol for her. Her partner had tragically died. He'd drowned several years before we met her, and she offered to make us this dragonfly for our group. And the dragonfly's body is like a double helix, a DNA image to really sort of represent that ongoing contribution to learning about familial AD. And so I think that is really very clear in the two studies that I want to share today. So we just learned this study was funded 2 weeks ago, didn't we, Nick? And it's incredibly exciting, very early stage. And so the title of this study is Antisense Oligonucleotides as Therapeutics for Autosomal Dominant Alzheimer's Disease. You can see why it needs a catchy little acronym, and you'll see from the acronyms today that whenever we write a grant application, we come up with these long titles, and the funders make us put a little acronym that goes with it. So
I'm sure the acronym is what determines the success.
Well, I think it might be actually, maybe not in this case. So oligonucleotides, antisense oligonucleotides, or ASOs as they're easier to say, are a type of genetic therapy, and we've heard a little bit about some of them which are in clinical trials that some people in this room are receiving. This is a very new type actually, so the types that we've heard about before target the protein APP, or the amyloid or the tau protein directly. This study is about actually targeting a little bit of the DNA material that is just before the presenilin-1 gene or the APP gene, and which will actually aim to silence that whole presenilin-1 gene, so that instead of producing the presenilin-1 protein that carries the fault and that leads to the production of these longer amyloid beta species, you'll just have the presenilin gene that was inherited from your other parent that doesn't have the mutation. So it's a really new technology, and what's really exciting is that it's being developed in the cells that come from people who've donated skin biopsies for our research. So many of you will recognise Selena Ray and Charlie Arbor if you've been coming to these meetings over the last few years. So these are the incredible cell biology scientists that we collaborate with. We've been collaborating with since 2009, I think. Selena gave a talk at our 2011 meeting when she just started the study, and one of our participants got up and showed her arm and said, "I've just had a biopsy, it was fine. Natalie did it very well." She was very complimentary, and lived to tell the tale about it, and really got everybody involved in donating skin biopsies. So from the fibroblasts, which are your skin cells, they have this amazing technology which just never ceases to blow my mind when I talk about it, that they can, by growing it in a special soup, turn those cells into induced pluripotent stem cells, which were the cells in the body that could have become anything. And then by growing them in a different soup, and Selena will forgive me for using this language, which is not the language she would have used, but they grow patient-derived neurons. So they can actually look at what is the effect of the mutation in the cell type that comes from somebody carrying it. And actually they've been really welcoming to Briony. They invited to her lab so that she could see some of the work they do, and that's what gave Briony this idea of framing some of the family photos in Petri dishes to really connect scientists in the building with the people who've donated these samples. So we've also had a very long-standing collaboration with Bart de Strooper, who is a very eminent scientist based in Belgium and also the UK. He was head of the UK Dementia Research Institute. He's done really groundbreaking work looking at the mechanisms by which these mutations cause disease. And so he's teamed up with us, with neurologists working with families across Belgium and in the US. And he's just been funded with a very big grant from the VIB, which is a Belgian research institute, to develop this gene therapy in cell models, with the idea that if it's effective, we could then start thinking about really personalised therapy. So it's early stage, it's at a preclinical, so a pre-person level, but I just love the fact that it's using the things that people in this room and their relatives have donated, and you'll be hearing lots more about this research as it develops over the coming years. Obviously, these kinds of therapies, to be really effective, we would want to give them early. So we would want to give them at a stage before too much damage has taken place. And so I think they also highlight how important it is that we have better ways of detecting the very earliest stages of the disease, which leads into the other study that I wanted to talk about today, which I couldn't resist the acronym DRAGONFLY for this one, to really acknowledge everyone who's contributed. So this is detecting cerebral amyloid angiopathy and familial AD to reduce the risks of amyloid immunotherapy. So what is CAA, or cerebral amyloid angiopathy? Well, it's a really important part of Alzheimer's disease pathology that we maybe don't talk about so much, as we know about the amyloid plaques, we know about the tau tangles, but actually most people with Alzheimer's disease also have some degree of amyloid deposition in the walls of blood vessels in the brain, and this is called CAA. And so it's present in probably about 80% of people, and this is based on post-mortem studies, and is why post-mortem studies are so important. But what we've seen by working with brain tissue from people with familial AD is that the severity really varies. So 2 people could both have very late stage Alzheimer's disease, but have a great difference in whether they had, and we can see here, this is a blood vessel with really no amyloid in the blood vessel walls. This one's mild, this one's moderate, and this one's really quite severe. And so, didn't mean to do that. So we need to be able to detect this better during life, and the reason why is that we know that the risks of amyloid immunotherapies are much higher if you have more severe CAA, because your blood vessels are already quite fragile when you've got an antibody coming into the system to remove amyloid. And we do have some what we call structural brain imaging markers of CAA. So over here you can see these little black dots. I don't know if you can see, these are microbleeds. So these are what Nick was talking about earlier, tiny areas of bleeding that don't cause any symptoms. These are one of the things that can develop on amyloid immunotherapy, but they also occur in about 1/4 of people with familial Alzheimer's disease without therapy, and they don't cause any problems. They're just a sign that there is CAA there and that it's causing these effects. And some people, this is an old brain hemorrhage. They're quite rare in familial AD actually, but sometimes you see signs that there's been a small bleed in the brain, again, which might not have caused any symptoms. And these are white matter hyperintensities, are another sign, but they're very non-specific. You can get these white matter hyperintensities just with aging or with other inflammatory conditions. And so all of these markers that we can detect already, and that we do scan sequences for when people have an MRI scan, they're picking up the results of CAA, so the damage that is occurring in the brain. But we know that that must be preceded by many years of alterations to blood vessel function. So what we want to do is be able to pick up these alterations in blood vessel function before they cause damage, because that's what we want to be preventing when people have early treatments. So we need to be able to detect these earlier changes. Okay, so to talk about how we're going to do that, I need to tell you a little bit about the biology of cerebral blood flow, which is really amazing actually. So our brains are incredible in so many ways, and one of the ways they're incredible is that when an area of our brain gets more active, the neurons, the nerve cells, are really tightly coupled to the blood vessels in that region. So you get this increase in blood flow. This is from a mouse who's having his whiskers tickled, and as soon as his whiskers are tickled, that tickle area of his brain is saying, come on, blood flow, I need more oxygen here, I need more energy. And so there's been a lot of science really trying to work out how you can measure that, and actually we can also measure it in life in a slightly different way, using an MRI scanner. So we've done a lot of structural MRI in our research studies, but we've done less of this technique that I'm going to talk about, which is called functional MRI. And so this measures something called the BOLD, which stands for blood oxygen level dependent MRI, and it does so when someone's doing a task. So in this task, somebody lying in the scanner is looking at this rotating checkerboard. So that's a very visually stimulating thing to see, and so the neurons in the back of the brain that control vision get very active, and they signal to the blood vessels that they need more energy at the back of the brain. And interestingly, for reasons we don't quite understand, CAA particularly affects blood vessels at the back of the brain. So in this study, to detect early signs of blood vessel dysfunction, we're going to be asking people to lie in the scanner, and after they've had all those structural sequences, we're going to be showing them images like this, asking them to press a button on the screen when they see them, and looking at the blood flow in that area of the brain. And we already know that this can detect very early signs of CAA, and we know this because of another incredible group of people who are affected by a rare genetic condition. This is people with HCWAD, also known as hereditary cerebral hemorrhage with angiopathy of the Dutch type, or Dutch CAA. So this is a very, very rare mutation in the APP gene, which interestingly doesn't cause Alzheimer's pathology, it causes pure CAA, and it affects families they mainly come from 2 coastal areas in the Netherlands, and they've been incredibly active in participating in research, and I had the privilege of meeting some of them last year at the International CAA Conference. They've also set up a foundation that led to the formation of this conference that brings together scientists working on CAA. And this is from a study where they were doing this exact task in the scanner, and the blue line shows the people who carry a mutation, and this is really looking over time. This is looking at that reactivity, the reactivity of the blood vessels. So they're just slower, and they're a bit stiffer because of the amyloid in them. And at a group level, that's been detectable. And actually, this type of sequence is being used as an outcome measure in some clinical trials of a gene therapy that's specifically targeting the APP gene in this cohort. And so it's a little bit more complicated in familial Alzheimer's because we know there isn't just a blood vessel problem. It's a bit of a chicken or the egg situation. So we want to know what comes first. Is the amyloid in the blood vessels making the blood vessels sort of slightly less reactive, or is it the amyloid that's affecting the neurons that are signaling to the blood vessels, making them less reactive? So what we're also going to bring into this study is magnetoencephalography, to measure the activity of neurons. And some people have already had the pleasure of having a MEG scan for one of our other studies, which involves sitting in these 1950s hairdresser style machine, but instead of coming out with a perm, you come out with the science of having a really incredible readout of brainwave activity. And then with some really clever maths, we can disentangle the neuronal from blood vessel effects. And by we, I mean the royal we. And this is very much a collaborative study. Nick and I collaborating with two really incredible and really lovely scientists. So Professor Martina Callahan, who's a professor of physics, who leads the functional imaging laboratory, which is just next door to the Dementia Research Centre. And anyone who's been participating in research recently will know that we now do our MRI scans there. So this was partly due to an unfortunate event that happened to the hospital scanner, which meant that it's out of action now, but actually we were wanting to move to scanning next door anyway because we're building this collaboration. And then Peter Zimmermann in the middle is a professor of neurovascular modelling, so this is literally what he works on. He uses maths and computer science to understand the signals that come from these two different approaches, and he kindly put together some of these slides. He's the kind of person, when you hear a talk, and he was talking about his work looking at neurovascular modelling, and I just went up to him afterwards and said, well, you have to work with us and you have to work on this study because your cleverness needs to be put to address this problem. And he's been absolutely brilliant to collaborate with, and we're really excited that we've got some funding, and so we're going to start piloting this study, hopefully starting later this year or early next year. And that's it. I'm happy to talk to anyone who's interested in finding out more at the end, but want to thank everyone on the research team who's going to be involved in this, our collaborators on this particular studies, and everyone who takes part, who's sort of led to this point and continues to shape the research we do. And we're going to have an Alzheimer's Research UK funded PhD student working on this with us, and so I'm sure you'll get an opportunity to meet her next year. Thank you. Gone completely over time, which actually I don't think was entirely my fault. I think we were slow to start. We're going to have questions at the end as part of the panel, but I'm really delighted to welcome Chris Hardy to give the next talk. I will let you introduce yourself, but it's just been so wonderful to have you join the team, and I know you will have met some of the people in this room. You're going to talk about your funding for your fellowship, aren't you?
Yes.
Great. Okay, because I think that is a very poignant moment for everyone to hear about that.
Yeah, thank you. Thank you very much, Natalie. Thanks for the invitation to talk today, and I do recognise many of you have taken part in this study already, so it's nice to see some familiar faces. I joined the long list of scientists who have a very tenuous acronym explaining AUDITION stands for Auditory Biomarkers and Dementia, Improving Diagnosis and Tracking Disease Evolution, which is but I'm really interested in the relationship between hearing and dementia. And I'll start talking about why that relationship is interesting broadly in the context of dementia, and then I'll come to talk about it specifically in FAD. But very broadly, there is this nebulous relationship between hearing impairment and dementia. And I don't think it's fair to say that we understand the nature of that relationship just yet. It is a relationship that you might have heard about. These are a carefully selected, relatively balanced spectrum of political newspapers highlighting the nature of that linkage. And I think there are three main theories as to how these two things could be linked. So the first theory is that hearing changes in the ear somehow cause a neurodegenerative pathology like Alzheimer's disease. The second theory is that it's not such a direct linkage, that perhaps changes in the ear are linked to dementia through a different mechanism. So perhaps if you start to lose your hearing as you get older, you might not socialise so much, and we know that social connectedness can be protective against the onset of dementia, so perhaps there's that indirect linkage. Or perhaps if you lose your hearing, you're devoting more of your cognitive resources to focusing on speech and noisy environments, and perhaps that could unmask a dementia process earlier. The theory that I really work on is the idea that we might have some of this relationship the wrong way round, and actually rather than hearing impairment causing dementia, it's that dementia might cause some changes in hearing. The reason I think this has some sort of face validity, why I think it's a reasonable hypothesis, a reasonable prediction, is that actually this is an image I drew for a paper about 10 years ago. You can see I'm not I'm not an artist, I'm not a scientist. But here I try to colour in the different regions of the brain that are involved in hearing, and the different colours, don't worry about the different colours. The point I want to make with this map, so-called map, is that actually we hear not just with our ears, we hear with our brains as well, and huge swathes of the brain, these huge brain networks are involved in hearing. On the other side, we've got this another very crude map of what I've called dementia, and again I've coloured in different brain regions representing different forms of dementia. And really the only sort of take-home message I want to get across from this slide is actually, if you look across those two maps, I think there's huge overlap, and you can see that I think it's reasonable to predict that people who have early changes in the brain due to a dementia might have early changes in hearing caused by those changes in the brain. So I've sort of introduced this concept of brain hearing, and I want to try and explain a bit more about what I mean by that. So this is a conversation I had with a research participant a couple of weeks ago. There's nobody in this room, I've checked that. It was very instructive for me in terms of how I explain this concept. And I started with this question: do you have any problems with your hearing? And when I started research 10 years ago, I would have probably stopped at this question. I said, do you have any problems with your hearing? And the gentleman said, no, actually I think my hearing is pretty good. But so I said, okay, well that's great. And what about in situations, where there's lots of background noise, like in a busy pub or a restaurant? And he said, oh no, well that's the bane of my life. I can't hear a thing, especially if there's music playing as well. I used to enjoy going out. So we went on. I said, I'm sorry to hear that. And what about, if you're trying to hear somebody on the telephone? And he said, well actually I find that quite difficult now too, because I can't see the person's lips moving. And it's even worse if they have an accent that I'm not used to. And I asked him one more question, which was, is there anything else that you've noticed about listening to certain sounds or music or anything like that? And he said, well, I don't think this is actually hearing as such, but I can't stand certain noises anymore. Things like a baby crying are really physically painful to me. And I would argue that all of those things are types of hearing that we don't necessarily think of as hearing. And research shows that people with typical Alzheimer's disease in particular really find situations where there's a lot of background noise, like in a pub or a restaurant, very difficult. And that's a situation which we all find difficult. It's difficult to hear, but the critical thing is it's disproportionately difficult if you've got that dementia. So I'm going to try. Sorry, spoiler. I'm going to try and give an example of brain hearing in action. So this might work, this might not. So I'm going to play you a sentence that I have drastically distorted. It's going to sound very strange. You've just had a sneak preview of that. Some of you in the audience will have participated in the research. You might have had some priming to this, so you might actually be able to understand this. I can see a few people. But in theory, you shouldn't be able to understand the sentence until I play you the real sentence, the undistorted version. And when I play you the distorted version again, you should have a pretty good. I predict that most people in the room should be able to understand that sentence, and that, I would argue, is an example of your brains rapidly adjusting to the information that I'm giving you. So let's see.
Think how quickly your brain is able to adapt. Isn't that incredible?
So could anyone understand that? Any participants? Very good. Demonstration. Okay, so here is the actual sentence. I try to play it.
See how quickly your brain is able to adapt. Isn't that incredible?
I should say that these pictures, by the way, are visual representations of the speech signal. The bottom one is how the speech signal normally looks. The top one is how it looks after I've applied this distortion to it. So now, let's test this. So if I play the top one, this distorted version again, let's see if it's any easier to understand. Good. So I see some murmuring, some nodding. So I think that's a really nice demonstration of our brains able to very quickly adjust to the new information that I've presented you with. But I can't come to the next slide. Okay. So how is this relevant to familial Alzheimer's disease? Well, I don't think this has ever been studied except for this one paper published back in 2009 from a group in America. What they did was they tried to measure the electrical brain activity in response to different types of sounds that were played to FAD gene carriers and non-carriers before the onset of symptoms. What they showed in this graph is really that there was a difference in terms of how long the brain activity was taking to process these sounds. So it seems like there's a clue that there might be something worth looking at here. So what happens during the audition study? It takes around 45 to 60 minutes. It can sometimes usually be combined with other research, and it works really well if you're coming in for the main FAD study run by Nick and Natalie and the team. If you are short on time for that, we can do it in sort of 15 to 20 minutes in person and the rest online. In person, we would just need to do a measure of your hearing in your ear and a reaction time task. In total, you'll do 6 tasks with us, looking at different types of hearing with me or Ben, who is currently at Glastonbury. Who thought that was a better use of his time than coming to see this talk today, which I'm not sure. And we'll ask you some questions about your hearing as well. And it's all organised by the amazing Emily, who you all know. And it's a one-off, so we don't do this every year. It's just the one-off. So if you have a horrible time, which I don't think you will, so I think if you talk to people who've taken part, I think people mainly enjoy it. But if you don't like it, you don't ever have to do it again. What's the point? So I think this is one of the most important studies of hearing and dementia that's ever been undertaken, and that's very little to do with me or the researchers. It's all to do with you and the amazing contributions that all of you make to research, and the unique and special contributions that you give. I think it will help us to identify whether or not brain hearing tests can actually predict whether somebody is close to developing symptoms of Alzheimer's disease, and that could be very useful for things in the wider Alzheimer's spectrum. People come to audiology clinics and identifying people who might be at risk within a couple of years. And it could also pave the way for auditory rehabilitation strategies that might help people to manage in busy pubs and restaurants, and might help people to keep going to those social occasions that they enjoy going to, but often report not being able to cope with anymore. So I'll leave it there. I just want to say thank you very much to everyone. I think I've seen 26 people so far for this research. It was amazing. Thank you to the whole team, and also particular thank you to Stuart and Carol Jennings. I'm funded by the Alzheimer's Society, and about 6 months into my research fellowship, I got an email from Alzheimer's Society saying that my fellowship had been chosen by the Jennings family to be named in Carol Jennings' honour, and that was one of the best emails I've ever received. So thank you very much to Stuart and the family as well. Thank you all for listening.
Embarrass Chris by saying he's also just been awarded Trellisard Arbor project funding for the next round of art science collaborations. So. Okay, I'd like to welcome the wonderful Nikki Zimmermann and Ollie Hayes to the stage to tell us some updates on Rare Dementia support services and interventions.
As you see, for all of my talks today, I've picked somebody really tall to stand next to.
I'm just security.
Thank you. It's always lovely to come to these seminars, and this one especially. And just to give you an update of what we're doing at Rare Dementia Support. And this really sort of will blend in with what Ollie's going to talk about, the interventions. We work very closely together. I'm the PPI lead for all the recruitment, the RD talk do, and it works so well with getting to know our members and being able to funnel them into some really interesting parts of research, which hopefully will be able to turn into services as well. So I'm just going to bamboozle you with lots of figures. There will not be an exam later on these, I'll just let you know. But it just sort of shows sort of how busy we are. We come to these seminars, these meetings, and there's about 50 people in the room, and that's just a little bit of what we do. We have 7 different types of rare dementias that we support, and this is figures from May, which are now over 8,000. We've got a very small team, but there's 8,000 people on our membership. They're people like you, people at risk, people that are living with a rare dementia, people that are caring for somebody with a rare dementia, some bereaved carers, and also health professionals that are helping people out in the community as well. Our membership goes up every year by about 27%, and that's fantastic. You know, it's awareness raising that goes out there into the community, into health and social care teams, which hopefully will provide more information and more support for people. We do a lot of our support either through telephone or through Zooms. It's going to be so wonderful when we've got the centre and we can see people face to face. They can come straight over from their appointments to see us, or from their research visits as well. The website, we get lots of visitors to our website, and I had a sneaky peek at the tree out there, and somebody said, please improve the website. We are, honestly. It's in the process. It's a really, really big overhaul we're doing, but we will have a spanking brand new website soon. We're doing lots of the Many Faces of Dementia, the training. Again, I noticed somebody put talks and training on there. So as well as the training that we do online, we have fantastic champions and cascaders, which are our members, people like you. Again, they could be living with a rare dementia, a carer, bereaved carer, or at risk, and they come out either online or in person, either on their own or with me or one of the team into the community. And we've been doing lots of talks, raising awareness, but changing practice as well. We do lots of talks, especially, again, that was noted out there, in care homes, when people transition to go and live in care homes. And, you know, they're not used to seeing younger people in care homes, or they're not used to seeing people with non-memory type dementias in care homes. So we do a lot of work with that. So I'm going to get rid of all these data and talk a little bit about the services that we've got at the moment. So we have annual seminars like these for all the rare different types, but we also have quarterly carer seminars. So the carers only seminars, it's all about caring and support that carers need and information. So it's not about any of the typical clinical or research sort of talks that you would have within those. We have our virtual support clinic, so that's one-to-one individual support or family support. We can't possibly do a helpline, we can't manage that, but what we do have is clinic days every day where the support workers can provide support for people. We've got peer support groups, so there is a Familial Alzheimer's disease quarterly peer support group, which Natalie and Helen facilitate, and me and Ollie pop in and help when we can as well. And then we have monthly support groups peer support groups for people living with young onset Alzheimer's disease, and for carers as well, separate ones. We've got some online educational programs which are facilitated by our team members, and then we have different focus groups and PPI groups. So according to sort of what the needs are for using those groups, sometimes we have a monthly group where some of our researchers take their grant proposals, their projects to get some feedback. These guys are brutally honest, by the way. You were there this week, weren't you? Yes, they don't like it, they let you know about it. But it's a really good way of getting the research out there into our members and actually being able to feed them back. One of our colleagues, James Cole, who does a lot of work with MRI scans, and he took one of his little projects at the very beginning to this group, and then he's come back so many times. And he came back a few weeks ago just to inform them that there's now 4 of these fantastic scanners around the country, which the group were just delighted to hear that they've been a little part of that, and then decided that they needed to do a visit to it. But they used to have an MRI scan, so it would be better if I went in it rather than they did, which I'm not sure we're going to go through with that one. We have cultural programs as well, so we have lots of different art sort of activities that go on. Charlie Harrison's our lead for that, and he's working all over the place, basically bringing art to our members in various different formats, and that's going to be a really big part of the centre as well. And then, as I said, we work very closely with the research programme, so giving our members the opportunities to get involved in various different research. And then our members involved as RDS champions, so sometimes we call them cascaders, and that's telling their story, their lived experience, and that's really, really helpful for health and social care professionals, for community groups, golf clubs, places of worship, all of those sorts of things, because your voices are so much more bigger and important than our voices. So we've got 30 champions at the moment, and we're going to be working to increase that as we go along. So this is just part of the activity of what we do at the moment at the top in the white. So that's sort of what our service is at the moment. And then we're going to have the RDS Centre opening beginning of next year, which we're very excited about. And these are the sorts of things that we're going to be looking to introduce. So drop-in sessions, like I said, we're going to be 5 minutes walk from the clinic, from the Dementia Research Centre. People can come in. We're going to have some social events going on in there, some cultural events. We're going to be doing lots of teaching, so we're going to be developing MSc and postgraduate certificates. So in the next couple of years, those are going to be introduced, working with people in the centre, and some professional courses as well. We want to have some places where you can come and try gadgets and things, so sort of working with rehab and sort of assisted technology. We're going to be, we've got a small counselling service at the moment, but we're going to be able to expand on that. We're going to have opportunities for people to do some befriending and volunteering in there, so it's hopefully going to be a hive of activity, so we're going to need lots of people volunteering their time to just be that friendly face when people come in. And I think what's really important for this group as well is young family and children support. So we're looking at what different services we can bring in, and lots of sort of nice sort of art therapy-like sort of services to bring into the centre as well. And obviously, it's going to be part of the research. We want nice research in there. You're not going to come in there and get blood taken from you, or lumbar punctures or anything like that. It's just going to be the nice thing.
Maybe.
We might be keeping Nick out for that
If you
one.
see Nick lurking around the corner with a syringe, turn down the other one.
So just a couple of visuals of the centre. So it's 2 townhouses on Brunswick Square. The builders are in at the moment, and they're making huge progress. You actually sort of seeing now with walls coming down and going back at different partitions. I think for a while we went in and it was sort of like, please use your imagination, think really hard, whereas now it is actually taking shape. Just an array of different sort of activities and groups that will be going on. And then there's a picture of the Chelsea Flower Show garden that we had a couple of years ago. So the outside space we're going to sort of develop very similar to that. And here's just a couple of pictures of what we sort of see would be the inside. So this kitchen table is going to be the main hub. When people come in, it's not what's your name and what's your diagnosis. It's going to be come in, tea or coffee, come and sit down, and come and relax. And this is what we sort of see as one of the family sort of counselling rooms or support rooms as well. So we want it nice and homely, nice and bright, but just really welcoming for people. Right, I'm now going to swiftly go on to Ollie, who's going to talk about the trial that we're involved in.
Thank you, Nikki. It's a very good segue talking about support, because this is something that we hope can be an axis of support for you. We heard a lot this morning about the bravery of the people in this room and about these new senses of hope that are arising. And the work that we do in my line of work is help you connect with those emotions, help you connect with the positive, living alongside what can be such a difficult challenge. So as it says on the screen, we've got another acronym here. This one is actually pretty good. We've got the first letter on most of the words, so taking a little further in my cap, not throwing any shade over here. Improving well-being associated with rare familial dementias. That's what we're trying to do. So if I start us off, I'm just going to give us a little overview of what it is. It's an online course aimed at promoting well-being for people within families affected by familial dementias. And it's really important that it's framed that way to us, because that is our primary goal, to promote your well-being, to get you to live the best versions of your lives that you can, rather than focusing on reducing anything or focusing just in on your association with the familial dementia. We go beyond. We want to look at how to get the most out of life in general. There are 10 online modules, 6 topic areas, and you get 3 check-in sessions with a facilitator moving through. Each section is made up of videos, exercises, worksheets. We've got videos from the lovely Natalie Ryan in there, and a variety of other healthcare professionals as well. And each section is only about 2 hours of work, so it's not too much of a commitment at any one point. The program addresses connecting with your values, managing unhelpful thought patterns, living your life to the full, and the rather try and more at the bottom, because it's whatever you want to bring to it, really. That's just the structure that we lay out to try and help you guide yourself through. This is what the homepage looks like. Just to give you a little kind of sense of what we're talking about here. There are these videos which people in the room helped us with, and I'm looking at all of your faces. Thank you so much for all of your help in building it. And we've got lots of text around to help contextualize everything, make sure that nothing lands like a thud. Everything is contextualized properly, and it's a pick and choose. Anything that's useful, we encourage you to take with you. Anything that's not, leave it there and tell us about it as well. We always want to be getting feedback. This course is for people who know their genetic status, both if you're gene positive or gene negative, and people who don't. We know that about 75% of people choose not to find out their status, and this is a piece of research that you can get involved in, whether you know or not. And especially for people who are gene negative, something that we've heard a lot is that it can feel easy to get cut off from the support that you've been experiencing to that point. This is something that we'd really encourage your involvement in if you are interested. The course has specialized modules to address the different needs of different groups. For example, if you're gene positive, you'd go through a section about worrying about your symptoms or your status. If you're gene negative, you go through a section that deals with the topics of survivor guilt and anger. And again, I just want to keep reminding throughout, you don't have to connect to all of this, but it's just there for you if you do. So, down to the boring recruitment-y stuff. We'll make you do some questionnaires before you get involved. Once you get through the questionnaires, as I said, it's about 8 weeks, 3 check-in sessions throughout the 8 weeks, and then we make you do more questionnaires again, because we love a questionnaire. We currently have about 35 people recruited. I don't know if I've mentioned yet, but this study has existed in frontotemporal dementia for about 9 months now. And during those 9 months, we've been working really hard with a group of people within familial Alzheimer's disease families to adapt it properly to make sure it meets your needs as well. We didn't want to be careless and assume similarity. So, you're late to the starting line, but you can build up your own numbers very quickly if you are interested. Please do get in touch if you want to be a part of it. Now, it's a randomized control trial. I know you're all familiar with this structure now, so I won't spend too much time explaining it, but what that means is that 59 people, half the people who sign up for our study, will receive access to the program immediately. The other half will have to wait 6 months before receiving access. A little methodological quirk that we need to fulfill. So this is some of the feedback from frontotemporal dementia. Just to kind of highlight a little bit about what we're trying to do here, that it is helpful, and as you see in the bottom quote, it extends beyond your relationship with dementia. It can go well beyond that, in fact. So it's something that I would encourage you to think about no matter where you are in your journey at the moment. I've kind of alluded to this a lot already, but it's something that we've worked really hard on to make sure that we are representing you all in this. And in our design team, We had people who were gene negative, people who were gene positive, and people who didn't know their genetic status. So you've all been represented in there, and I hope the people who were in this room who were a part of it would agree it was a process that we made sure that we were collaborating on each specific section and moving through and rechecking our work with each other. Doing that work together made us make some big changes to the course, and this is something that has also been a symptom of our work on this intervention, is infinite change and reconstruction to get it to the right place. So it's not something I'm bringing to you lightly. Engaging with your relationship with your risk and dementia within your family is not something we've taken lightly at all. So, after all of that drawing on, if you feel this would be a good fit for you or someone in your family, or someone in your family, it doesn't have to be one or the other, please do sign up as a Rare Dementia Support member at first, which I suppose you all have, hopefully.
Absolutely.
Yeah,
No.
If not, stay after class. And then get in touch at the contact@raredementiasupport.org email address. Thank you for your attention.
Now it's my absolute pleasure to welcome Seb and most importantly Tamara to the stage to talk about creativity in rare dementias and a very exciting new project on the horizon.
Hello. I'm going to come up here because otherwise I'm going to be one of the shorts. It's a real pleasure to have you with us today. And for anyone who's been sitting in the audience wondering what this is all about, this sort of ghostly figure behind Chris and Natalie as they were speaking, you're about to make sense of it. Just before I ask you to introduce yourself, people will have heard your name mentioned by Briony in relation to the Walking in Your Footsteps project.
Yes.
And your wonderful dad and his beach mazes and things. And I wonder if you just wanted to start there by saying a little bit about your connection with that fantastic project.
Yes, well, thank you for having me. And yes, it was a real honour and such a privilege to go to Margate with this amazing group of people, and it was an amazing context to be in to kind of connect with people. I think we're in a sort of non-clinical setting and kind of really build community around ourselves, and we had a right laugh. We really did, and I just sort of saw such kindness and resilience. My dog got stuck in the car, that was a whole other thing, and everyone like rallied round. And it was, very magical. And I sort of really saw the value in that sort of thing, doing a creative pursuit together has real value, I think. One participant was like, oh yeah, this is great, because in my area all they offer is knitting. And no shade on knitting, which is great. But she happened to want to do sort of different artistic things, and so I really hope that we can carry on doing something like that again. Yes, and that sort of having that community around has kind of given me confidence a bit to start working on my background is that I'm a performer, and yes, my dad was one of the first DIAN patients. He had early onset Alzheimer's. I'm living at risk but don't know my genetic status. And yes, I thought I'm going to try and start making a show that has sort of clowning cabaret elements to it. So I'm going to share a very short extract from the beginnings of it, and welcome critical feedback. Yes, one thing to say is that one of the people that slightly inspired this was, I know there are very many wonderful carers in the world, some of whom looked after my dad. There was one lady who came in to the room when my dad was, on his deathbed, and she came in and she put her hand on my shoulder and she went, I'd never met her before, she went, "Time's a great healer." And I thought, okay, I'm going to find
find that sort of darkly funny in the future, I hope. So I wanted to say, yes, one of the characters you're about to see is based on her.
And so just before you give us a little taster of Forget-Me-Not, we often talk about the kind of projects that Natalie and Briony and you and others here have been involved in as being like this fantastic triangular connection of people with lived experience and scientists and artists. And obviously you wear clearly at least two of those hats. Is there anything else, either with your lived experience hat on or your artist hat on, that you want to share with people to sort of help them understand what they're about to see, or as a little primer?
Well, gosh, I know that we're cutting into tea time, so I don't. But yes, I think. Well, I find theatre happens to be very cathartic for me. I know it's not for everyone, but I hope that it's helpful to sort of see. I'd love to share stories with people and for their stories to feed into this. Yes, I'm not sort of playing myself in, as it were, and I'm trying to sort of represent other people's stories, and I think, coming together and doing theatre stuff together can really build resilience to face that tough stuff, and hopefully to find little glimmers of hope amongst the darkness. But because the stories so often, it's obviously so much loss and so much sadness, but I see in everyone I've met such willingness for humour and little flickers of hope and joy along the way.
Well, it's a chance that we literally have a stage here, but I'm going to vacate it and hand over to you.
Okay, it's just 10 minutes. Hello everybody, welcome to Forget-Me-Not Residential Home. Thank you for coming to our cabaret afternoon. Welcome residents and their families, future residents, and a special thank you to our sponsors, Better Place Funeral Care. Okay, so my name's Jane, I'm a carer here. I'm here to care for all of you. Oh, don't we all look lovely? Haven't you scrubbed up nice, fabulous? Oh, this is lovely, you've really made an effort. Now, let's put our hands together for our first act, Sarah. Hi everybody, hi. My name's Sarah. My husband Paul, he's got early onset Alzheimer's. He's not a resident here, not yet. But yes, I just thought I should come and check it out, and I needed a moment to myself, quite frankly. And I saw you were doing this cabaret afternoon, and I thought, well, I could bake an Alzheimer's cake for you all. Every cabaret needs a cake, right? Gosh, is it hot in here? Right, so my Alzheimer's recipe first. You take 3 eggs. Paul and I have 3 kids. La da da yes, how to break the news to the children. I don't know. I mean, you can just crack on. Everyone's gonna get scrambled one way or another. What's the time, Lucy? It's 12:55, my darling. That's my husband, Paul. He's asking the time every 30 seconds at the moment. So, you take a nice big bag of free time and you sift it through the sieve until there's absolutely none left for yourself.
Fabulous. What's the time, Lucy? It's still 12:55, my darling. And then you take your future plans from the top shelf and you break them into a saucepan, ready to melt them on a low heat. Oh, don't forget to add 150 grams of anxiety. I always stick in a little extra myself. And melt that into a nice little puddle of worry. Leave to simmer. Okay, don't forget to just a spoonful of denial helps the medicine go down. The medicine go down. Medicine go down. In the most delightful way. What's the time, Lucy? It's 12:56, my darling. Now you put that mixture into the batter and whip it all up, and that's curdled. Well, we are where we are. Into the oven we go. Spunk. Now for Alzheimer's, one would usually set the timer for 80 years. But as we all know, you can set it at about 40 perhaps. Great little time-saving trick. And now for the sauce. Okay, so now we get a great big vat and you can put all your exhaustion into the vat. In fact, if anyone's got any exhaustion, just take it and chuck it in the vat. You can chuck it right at me if anyone's got some exhaustion. Oh, thank you. Oh, yeah. Oh, that's heavy. Any more exhaustion over there? Yes. Fabulous. Yes. That's brilliant. Thank you so much. Oh, that's heavy. Right. Then you grind in your frustration and just add a little pinch of resentment. I didn't think I had any, but then I checked right at the back of the cupboard. Oh, I've got plenty! Here it goes! More resentment! There we go! In it goes! Fantastic! What's the time, Lucy? It's 12:57 and I'm Sarah, your wife. Oh, I'm sorry. I'm sorry. Oh, a rich ganache of guilt. Oh, that's going to take more than a Gaviscon. Of course, I still love Paul. In a different way. He used to be very strict about his diet, you know. But he seems to enjoy his food now. He does keep leaving his shaving foam in the fridge. What's the time, Lucy? It's time to dance, my darling. Wise men say only fools rush in, but I can't help falling in love with you. Would it be a sin if I can't help falling in love with you? Like a river flows surely to the sea, darling. So it goes. Some things are meant to be. Take my hand. Take my whole life too.
things are meant to be. Take my hand, take my whole life too, for I can't help falling in love with you. Falling in love with you. Oh dear, don't worry, it's all right. Tears are just pain leaving the body. Oh dear, oh. If anyone's got any issues, Jane's here with some tissues. Thank you.
Okay. My great belief in these meetings has been confirmed yet again, which is everybody prefers the coffee and the tea breaks, and they're the most important points. So we're going to just have a brief session here. It's a great pleasure and privilege that we've got Niamh Kennedy here, who's a documentary director. I'll tell you why she's here in just a moment, but I'll embarrass her by saying that she has been incredibly kind, coming all the way down from Edinburgh, especially for this. So thank you very much. And I'm going to introduce the other two in just a second, but how I met Niamh was through a documentary that it's award-winning, and Niamh will tell us a little bit more about that. A BBC Two documentary called Jennings versus Alzheimer's disease. And we have two of the characters who play a very key role in there. Stuart, who's Carol's husband, and John, who's Carol's son. And Carol, for those who don't know, you may have seen the letter earlier. That was Carol who wrote that letter to John Hardy saying, I think my family could be of help. And I still have that letter in my office because that letter was absolutely right. And a family could be of help. And that's how the first gene was found. And for those of you who were here for my introductory beginning, that led on to understanding proteins, the first transgenic mice models for Alzheimer's disease, the first treatments, and now to licensed therapy. So that's an amazing thing. But what I wanted to start, and why this session, you know? And the reason for it is because there is so much out there about misconceptions and ignorance and myth, and there's a lot we still need to do. And I've got Richard very kindly has given me permission to share something that he wrote to me recently, describing how when his wife was concerned about her cognition, and he was concerned, I think that's fair, Richard, isn't it? And was struggling a little bit at work, went to get help from their GP and then sought multiple other medical advice. And in the family there was a very clear early onset in 30s, in not just parents' generation, but the generation above that. And I won't describe how all the details that Richard and family went through, but let's say people kind of come from a point that it couldn't be young onset, it couldn't be that, and there must be some other explanation, and you can see where there's a natural tendency for that, but it's not an uncommon story that I've heard in clinic, and we need to change that. We need to change the fact that there is no point in making a diagnosis because there are no treatments, because there are treatments, and there is so much lack of awareness. And cancer is decades ahead of us in terms of, it came out of the closet decades ago about awareness, and that led to research. There's 20 times as much research in cancer than there is in dementia, and that leads to treatments. People sometimes say, wow, the treatment's been found in dementia. And the answer is, well, they are being found. And the reason there's so many treatments in cancer is because it didn't happen by chance. People worked hard. And there were many researchers and a lot of funding. And that's what we need to change. And a part of that is changing politicians' views, but it's also about changing the world's views, the general population, the GPs, the neurologists, the psychiatrists' views. And awareness is key to that. So it's really lucky that we have people like Niamh helping to bring those stories, because the stories that hopefully you'll be spreading awareness through families and things like that, which is great, but if you can reach large audiences, and we're going to ask Niamh about how you reach large audiences and how you get over messages in a moment. But John suggested I play a clip which I hadn't realised he'd picked out one which features me, so I apologise.
There's a bit of content warning as well, because it is quite confronting, this video, but it's one of like 3 that are on YouTube, so we made a selection.
So anyway, I'm embarrassed, but I will play it, and it's all John's fault. We'll hope to play it.
Now this is 2002, 2003, 6, 7, and so on. And only because we can compare it to an earlier one, do you start saying, well, look at, maybe a little bit more space now.
Yeah, for me that's the latest one where we thought potentially it could just be something else, but we were raising concerns in the hope that it wasn't.
And now if we just move on, 2010, all the way through 2016, can you see these spaces opening up? The black areas.
Yeah, these black areas, is that just space? Is that just fluid?
That's fluid.
Cerebrospinal fluid.
That's replaced where there was brain tissue. And
that's 2016.
That's 2016.
Between maybe this scan and this scan, we started raising concerns about the way that she was behaving, not remembering things, not wanting to engage so much, not initiating conversations, and that sort of thing.
And this kind of explains it a bit, doesn't it? You see
the year I got married.
Wow. But now if we show you the whole series so you can see the change. And that's what the disease does. It's devastating. All the cognitive functions, calculation, language, visuospatial, planning skills, that make us what we are as a thinking human, all being affected.
Just stop it there. Thank you very much. Shall I turn this off? We don't need it. Would you be able to do that, Seb, for us? Thank you very much, my wonderful assistant. Niamh, thank you very much for coming along and giving up your weekend, coming a long way. Can you tell me a little bit about how it came about and the journey of the film?
Yeah, sure. Well, it started off, often production companies have conversations with broadcasters and they tell them what they want, and I think it started out with a simple conversation that the production company Expectation was having with the BBC, and the head of documentary said, I'm really interested in the subject of Alzheimer's. Can you have a dig around and see if you can find, something that you can pitch to me on that? And there was a really brilliant development producer, Jess, who just started researching, and she discovered a long-form article in the Times about Carol's story and the Jennings story, and I think then just found John.
She sent me a message on Twitter in the days that I still had Twitter.
And I think, and John was very willing to chat, which is the first huge step when you're making a documentary, is to actually for people to agree just to have a conversation with you and to start that conversation. And after that, and once she had met John and met Stuart and got the family on board, the next step was to get UCL and UCLH on board, because I think we knew that it was a story not just about a family, but a bigger story about the whole research that's going on, and the fact that the family story intertwined so beautifully with the research and all the drug developments and all of that that's going on. It just felt like the perfect story to illustrate, that people hadn't seen before, what Nick said about challenging preconceptions of what Alzheimer's disease is. I think, we've seen a lot of documentaries about, carers and about that struggle and that kind of thing, but I felt like we hadn't seen this side of it as much.
A huge, really very fantastically important for awareness of all those other things, but this, because it had familial Alzheimer's disease at its absolutely front and centre, was fantastic, and it almost felt like a science documentary by stealth, because you did it. Stuart, what was it like being approached, and what were your feelings? Because you had Carol at quite a severely affected stage at that stage.
Yeah, it was in the final stages really when we were making.
And can people hear? Okay,
yeah,
great.
Yeah, it was in the final stages. One of the things that helped is that Carol was vocal about the disease right from the beginning. The letter you saw, the clip, was 1986. You sort of realise how long ago that was. When Carol was in the early stages, when we got the diagnosis of cognitive impairment, I used to go with her, and she used to say to me, we must continue to make the story known, because there's so many people. I mean, I feel as I've got family members all around the world. I wanted to honour Carol by keep telling the story, to get more and more people involved. But as a carer, I felt I wanted to protect her as well. So it was finding that right balance, and having met Niamh, I felt this was somebody we could trust.
I will say, when you mentioned the Times article, so that Times article that Jess saw and we hated it. We absolutely hated it. From the start, they didn't speak to us at all. The journalists didn't speak to us. They wrote about Mum's condition, and at that point, the only thing, if you searched her name, the only thing that came up was all the stuff that she'd done, the positive stuff that contributing to research. And the article went into quite a lot of work. It was horrendous, actually. It spoke about how ill she was, and it was talking about the fact she was in the late stages, and it had this horrible metaphor about her brain being twisted, and it used the word demented as well. I mean, it was literally the worst you can imagine. And so when Jess sent that message, she actually phoned me when I was walking home from work, and it was the opportunity to just rant.
She was holding the phone out.
And so I think what that did is it set the tone right from early on that we wanted people to listen to how we wanted this story to be presented. And it wasn't a story of like a tragic, look at this tragic person with dementia, because that wasn't the story at all. And I think what was balanced really well was the reality of what it was like being in our situation, but also just what a big impact you can have from the situation we found ourselves in. So it fixed that Times article situation.
He made compensation by Tom Whipple, the science journalist.
Oh, I don't know if you should have named it.
Well,
I haven't forgiven him.
He wrote an article just before the documentary was shown that was much more comprehensive and much more sensitive. And it was quite ironic, only a couple of weeks ago we heard that article won him an award.
You're far too forgiving.
And, it was kind of, I think that was one of the first things you told me when I came up to see you, was how much you hated that article.
No pressure.
It's mildly terrifying as a filmmaker, because in your head you feel the burden of the Jennings story, of John's story, of Stuart's story, Emily's story, also Nick and Kath and everyone involved giving up their time, and you're thinking, don't mess this up. This is, and I had come to it knowing very little about familial Alzheimer's disease, and I think there was a lot of pitfalls to fall into, and just I think the key starting out was just to be completely led by you guys, and to, before starting filming at all, just to open a dialogue with you and sit down and say, what is the film that you guys want to make? What is the story that you guys want to tell? But not just to start to do that at the beginning, but to keep that dialogue going throughout the process, because with documentary, you get curveballs and things change and things that you thought you might be following don't happen. And so I think
it's just that we're so determined for it not to be like tragic. You know, because none of us are tragic, like we're all here because we're like fighting this disease. It's not like we're all here to wallow, and so we wanted to make sure that there was, and you balanced that really well in the film.
Yeah, I mean, as a carer, I always approached it that I wanted to remember, celebrate what we had, not what we were losing. You know, and I felt that Niamh did that, well, the whole team did that really well. You know, there is an element of sadness, and it's terribly, but she would be so pleased that that letter in 1986 has brought us all together here in 2025.
And there were so many kind of points of beauty, I felt, within your story. Like, your dedication to Carol was just so profound and so powerful and so beautiful. Like, that, for me, that was kind of just one of the strongest things. And also with you, you know, your life is so full and so vibrant, and, you know, your relationship with Matt and how you guys were navigating that together as well felt really important to dig into. That you're not just looking at, you know, you're not just looking at the disease, but you're looking at your lives as a whole.
Can I ask you, John, what, you know, agreeing to something like this is sort of a risky thing in a way, because it's not like it's a niche thing. This is BBC Two prime time. You don't mind me mentioning, you're a teacher. That's, you know, children are sometimes not the most generous. What were your thoughts about, particularly, you were very open about being at risk?
Mm-hmm. Well, there's a couple of elements. I guess the first was the wanting to wrestle back the narrative after said article. Also, you know, so when Mum started getting ill like 20 years ago, basically, obviously I was 20 years younger. And it was difficult at that, it was difficult at that point to, you're so isolated because it's difficult to say, oh, you know, my mum's got Alzheimer's disease, because some people will immediately say, you know, oh, my granny loses her keys sometimes. And I wasn't in a situation where I was able to communicate with people about that. It was very, very isolating. And so I felt like it was important to show that that it's actually a disease, it's not just a condition of aging, but also that there's many phases of dementia, to refer to the little online course situation. I think that's really important. And you're right, you do make yourself vulnerable, but that's the way that you build connections. I will say that the kids who watched it, in fact, some of my colleagues actually showed it in school, if you can imagine.
I can't imagine.
But I think, like, you know, teenagers actually they are quite emotionally intelligent in some ways.
I think you know different ones.
No, I mean lots of them wanted to have conversations with me, and actually, I mean, the number of people who have come up to me in the most unlikely of places, like in clubs and in the street, and in fact I went to Hyde Park to see Kylie, which obviously was not where I was expecting to have some Alzheimer's conversations, but 3 separate people came up to me to tell me that I'd made them cry, which again isn't really what you want to hear, but like, yeah, it was
and that wasn't just your singing
nice. Obviously it had a huge reach. People wanted to talk about it, like, yeah.
I mean, the reach is amazing. I mean, I think it's very hard, isn't it, to really know the viewing figures, but how many people saw the film, as far as you know, and there's also lots of clips.
Yeah, so it was almost 1 million people watched it, which...
That's a lot of support group meetings, guys.
And for a single documentary on a terrestrial channel, that's these days that's really big figures. Like, you know, your Strictly Come Dancing and all that gets the 10 million, but for a documentary that was huge, that was really big, and especially, you know, for, you know, a documentary that's a science documentary as well, you know, usually you don't get those kind of viewings, and I think it got a big spread in the controversial times again, and it got a pick of the day across all the newspapers when it was being broadcast.
Again, embarrass me, it won an award. It was the best science documentary. Isn't that good? to do justice properly, we never do in these meetings, but what would the messages you'd have for people, either about thinking about being involved in media, but also about it more generally from your experience of all this, any of you?
There's nothing wrong with being vulnerable. It makes the story real, and we all feel vulnerable at times, whether we live in, either with the knowledge or with the uncertainty, whether as carers we live in with it or not. But you took me careful in the interview. Tell me this now, I'm not telling you the way.
Oh gosh. Unscripted.
You said, has it been worth the sacrifice? And it wasn't a sacrifice. It was a pleasure, it was a privilege. And I think those of us who are carers, we struggle, we get frustrated, we find it hard, but it's somebody we love. It's a privilege. That was a curveball.
I remember that. That was the very first time we sat down together, wasn't it? Yeah, that was really, really powerful.
And John, what would your message for people being involved in things like this?
Well, I mean, it was something similar, but obviously you said it much better. So. I think that article we had no control of because they didn't speak to us, but there were times when we have spoken to newspapers and they get little facts wrong or little details wrong, and because you're making yourself so vulnerable, there is a risk there. And I think because Mum was always so keen to have lots of publicity, we're just used to throwing ourselves at the will of some of these people, but it hasn't always gone well, so you need to tread a little bit with caution if you do want to speak out. And I think one thing, particularly if it's like local newspapers, you can say, well, I'd like to see a copy before you, and if they don't let you do that, then there's something
that's a red flag,
I would say.
Yeah, and I have conversations with, because people will often say, I've been approached about this documentary, what to do, and it's always a tricky one because when you're making a documentary, you can't ever give the people that are involved in the documentary editorial control, because that's usually the first thing people ask, can I have a decision about what goes on screen and what doesn't? And that always rests with the broadcaster, and they will never relinquish that because I suppose they can't. But so then it's about reputation and trust, and I always say to people, if you're approached, and I suppose that goes for like by a journalist or anything, I always say, first of all, look at what this person or this production company has made before. Does the tone of it sit right with you? How does their work, their previous work? Because people can come and they can sit down with you and promise you the world. In TV we can be very persuasive. And so it's about, and I think also it's about the gut feeling you have with that person on your first conversation with you. Like, do you feel listened to? Do you feel like they understand what you're saying? Do they take on board your concerns? Do you feel like they're just giving you platitudes and it's all those things, and it's a slow process, I think, of building trust. And I think that trust and that, the conversations should be ongoing throughout the process of whatever it is you're creating together. And it should feel collaborative rather than didactic, I think would be the...
Yeah, I'm only smiling because I was thinking about the times when Niamh approached me, and I feel like I always kind of gave you the third degree about, these are the bad experiences I've heard people have, and you mustn't do this, and you mustn't upset them in this way. But you kind of sort of listened to that and then moved on.
Yeah. Well, I think and I was thinking about this as we were watching this clip, and your advice was really, really helpful at times, and you helped me from kind of bungling things or going in too harsh, because like with the brain scans, I remember having asked Nick mentioned something about Carol scans being there and having them going back years, and as a filmmaker, when you hear that this archive exists, it's amazing. But then Nick said to me, but you need to think really carefully, because I said, like, could we show John them? And he said, well, you need to think really, really carefully about this, because this will be really, really emotional for John. And I think before I had that conversation with Nick, I hadn't considered how profound it would be for you and for your whole family to see those scans. And so that's where, like, I really needed somebody like you to guide me through that responsibly.
That's very dangerous if you're relying upon me to guide you responsibly. I think we probably should wrap them up there, but I think thank you, all three of you, for both for time now, but also for bringing something which is, people have previously said can't exist, or deny that it could be your wife's problem, to such wide attention. Thank you very much.